Another audience question here, Lena. I'm not sure if we got to this one or if you might have answered this one already. But what are your experiences using transient expression platform to make fast early decision regarding understanding product attributes? Lena or Meinhard, have we addressed that?
No, I think we haven't addressed that yet, the transient transfection. So a lot of companies doing that in their early screening. We also see that a lot of companies are doing that, but especially for, I would say for this complex proteins, I would say it's better to go to a stable pool stage than having only transient transfection available, because mostly these are totally different cell lines you are using for transient transfection in comparison to what you are using finally for the stable production. So, for example, for transient transfection, you're often using like hex cell lines, there's special hex cell lines, hex T cell lines, or also from Thermo XP CHO cell lines or XP hex cell lines available, which you can definitely use for transient transfection. But for doing a final and early decision, I would definitely recommend to use stable pools. And on that basis, you can definitely directly go into a single cell cloning and your clone development. So that is from my experience what I definitely would recommend.
Excellent. Thank you to our audience for these questions. A ton of great questions coming in. Keep them coming. Lena and Meinhard and Neeraj are here for another roughly 15 minutes. So if you got questions for them, bring them. I want to bring back the aggregation and degradation discussion for a minute. I've heard you all three of you refer to that as common shelf life limiting issues. But I don't know that we got too much into the meat of how those shelf life limiting issues are addressed. We talked about how they're identified. How can they be addressed? How are your companies addressing them? Neeraj, maybe I can start with you on that one.
Yeah, I think I can speak to it a little bit more from a later stage standpoint. Early stage maybe Lena can comment on it. So our way of sort of addressing these things is kind of understanding right from the get-go, like from the standpoint of clone selection standpoint, at least that's where we started, kind of understanding what is really triggering these mechanisms. So in our case, what we understood was that aggregation is really a big issue when it comes to media composition, for example. For our case, for our specific molecule. So we have run a tremendous amount of DOEs to really understand the impact and therefore adjust the composition to specific to our molecule to minimize it. And of course, it is still a limiting factor for us during the shelf life, but we have adequate shelf life currently to go to commercial. So you're never going to address completely because proteins by nature, there is going to be some level of aggregation. For example, if the company wants to go to a high concentration formulation, I'll take an example, and you're going to see more aggregation because you have, by nature, you have, proximity of the protein molecules and so forth. But so it is a combination of addressing at the upstream level what we have been doing, and also at the formulation stage, and also, of course, aggregation also tends to be higher when you are storing, for example, room temperature compared to 2 to 8. So you take into all these factors, starting from the media composition to cell culture process parameters to formulation and the storage conditions. That's kind of how we address to get to a state where we have a molecule that is stable enough to go to commercial. So
thanks, Neeraj. Meinhard, Lena, any additions to Neeraj's commentary there?
Yeah, maybe I think one comment would be here. So if you see a high propensity in aggregation or degradation of a molecule, let's say during a manufacturability assessment, that I think the best would be to solve this at the molecule design level first, right? unless, but if there is no more time for that, right, then you have to go into this journey of setting up a process that somehow is able to generate these molecules with all the measures then in place to have the levels of aggregation and truncation low. And as Archie mentioned, then of course then the formulation is also in such a case the drug product development a very important part. What type of excipients do you need? What physical state of a drug product you will have to have? Most likely then in such a case it will be a lyophilized formulation to stabilize most.
Perfect. Lena, did you have anything to add there?
I think the most things are already stated from the both. So I think, yeah, like Meinhard mentioned, so if it's really critical and you have a high percentage of aggregation, I definitely also would think about the molecule design, because you will have it at all stages from production process up to formulation finally, and shelf lifetime of the product. You will have an influence there. If it's like, I would say, a medium aggregation, you can definitely do a lot of things. So we, for that kind of products, we, for example, already implement a media screening DOE already after clone selection, because we have seen the same Neeraj have seen. So it's so important to have a good media clone combination in that part, which can definitely influence aggregation finally, and really put like a difference of 10% of lower aggregates on your product. And also the general longevity of the process can definitely influence that. So then you need to think of, okay, is it perhaps good for that part to shorten my process, productivity, your USP process, and have like lower aggregation and lower fragmentation finally? Is it worth to do that? Because you also need to think of, then you will also reduce your titer. But you will also reduce your titer when you need to reduce the aggregates finally in the DSP process. So that's really a thing you need to keep in mind finally. So you have the possibilities of media screening, of process parameter screening, of process shortening, but you need to think of which might be the best one to have a final good commercial process in place.
Perfect. Heather, I'm going to ask you if we could present our second polling question. We'll give our panelists a break here and instead ask a question of the audience. And that question is, at what stage is proteolytic protein degradation primarily addressed in your development efforts? Molecule design, cellular clone selection, cell culture, or downstream development? Once again, I'll ask my panelists if they have any guesses here. They're thinking, where are we going to place our bets? All three of them are probably thinking, this would have been a good one to make choose as many that apply as well. Lena, what are your thoughts?
My thoughts are a B, because generally, perhaps people have an idea of a good fusion, for example, fusion product, because of the possibilities they add together. But they will only see when they are producing it in the cell line if it really, works like that. If we see degradation.
Yeah. Meinhard?
Yeah, I think this really then depends on the molecule. I think just the recent case that we had with an interleukin fusion molecule, I think we had to fix a degradation problem triggered by metalloprotease at the level of the harvesting, where we had to add protease inhibitors like EDTA. So this was a special case. And in other cases, I think cell culture or cell and clone selection, of course, will be important areas where you would address this to avoid. I think the more degradation you have in this B and C area, right, that the more the downstream has done to work to get rid of those molecules and the lower the yield. So I think you can fix it with downstream, but of course then the yield and the efforts in downstream are much higher. So therefore, to have degradation reduced at the minimum level would be, of course, desired. And this can then only be done in BC area, right?
As soon as you mentioned the downstream implications, I saw Neeraj's head start to nod in furious agreement. He was like, "Yeah, let's try to avoid it. When you throw it over the wall, let's try to make this as clean as possible."
Yeah, I think, yeah, I agreed. Yeah, you don't want to get to the downstream and try to kind of take all that load and try to see if you can remove all those impurities. I would focus on, yeah, cell culture and, upstream and, even the clone selection. But yeah, at least at a minimum, cell culture process need to be really optimized to minimize this degradation.
Well, let's take a look at how the audience responded. We'll see if this is. Oh boy, that's not music to Neeraj's ears. He's he says no. I think we have a lot of early process folks on the call who are like, yeah, we're all too eager to throw it over the wall and let downstream to deal with it.